friends to a new session on genomics and proteomics. In this session, we will discuss family studies and complex traits genetic maps. Gene mapping is the process of finding the location of genes on each chromosome. It is an important step in identifying the genes involved in a genetic disease. Once a disease gene is accurately located, we can determine its DNA sequence and its protein product. For example, cystic fibrosis is the most common lethal inherited disease in the United States. As many as 1 in 2,500 Americans of Northern European descent carry a gene with CF. In 1985, the gene was mapped to chromosome 7q31q32 by linkage analysis. 4 years later, Francis Collin and colleagues determined its DNA sequence. Now we know that the disease is caused by the defect of a chloride channel encoded by the CFTR gene. Relative to linkage analysis and sequencing, genome-wide association studies have less power in cases where different rare mutations act in different families or individuals at the same locus, that is allelic heterogeneity. However, they are far more sensitive than family studies to complex polygenic associations in which a phenotype is associated with the joint effect of many weakly contributing variants across different loci, that is locus heterogeneity. In this sense, GWAS have been a resounding success, identifying thousands of disease associated loci and revealing previously unknown mechanisms for conditions such as Crohn's disease, type 2 diabetes and macular degeneration. Family studies. Family studies have been used in the genetics since its beginning. One of the most successful approaches to identify new genes responsible for a disease has been the search of the full genome in families of affected individuals. Mendelian ratios were based on a relationship between sweet peas. The offspring get their genetic information from the parents, so they can have different genes than them. That is a principle of family studies. Pedigree, also called family tree, is a graphic parallel to a family relationships. Family studies have been used to identify monogenetic diseases, highly penetrant traits of a biomedical importance, including a variety of inborn errors of metabolism and their genetic diseases. Example, cystic fibrosis, Duchenne muscular dystrophy and Huntington disease. The importance of family studies. Family studies have several favorable features for gene discovery. Studies of extended pedigrees or even nuclear families are likely to represent a more homogeneous and limited set of causative genes and pathways. Members of the family usually share not only genes but also the environment. So, if only one or more but not all members of the family are affected, we can exclude the external influences and recognize the origin of hereditary diseases. The special twin study case is a perfect example of how the environment influences the origin of the disease. On the other hand, distances is the major disadvantages of family studies. Sometimes it is more expensive to create a pedigree of affected families than the unrelated people because the relatives can live far away. There are some typical diseases which are connected with higher familiar occurrence: schizophrenia, bipolar disorder, suicides or eating disorders. The use of family trees. Genetic linkage, genetic mapping, finding the origin of disease. Complex traits genetic maps. Genome-wide association studies of common complex or multifactorial diseases have been spectacularly successful in the last 2 years. Variation in gene expression is an important mechanism underlying susceptibility to complex disease. The simultaneous genome-wide assay of gene expression and genetic variation allows the mapping of the genetic factors that underpin individual differences in quantitative levels of expression. Expression quantitative trait loci or eQTL. The availability of systematically generated eQTL information may provide immediate insight into a biological base for disease associations identified through genome-wide association studies and can help to identify networks of genes involved in disease pathogenesis. The functional effects of DNA polymorphism on multifactorial disease may be mediated through several mechanisms. Polymorphisms that alter protein function can have very important effects. For example, CARD15 NOD2 mutations in inflammatory bowel disease and FLG mutations in eczema atopic dermatitis. The systematic study of complex diseases with non-synonymous SNPs has not in general yielded highly significant results, and variation in gene expression is probably a more important mechanism underlying susceptibility to complex disease. The combination of whole genome genetic association studies and the measurement of global gene expression allow the systematic identification of eQTL. The eQTL maps provide an important source of reference for categorizing both cis and trans effects of disease associated with SNPs on gene expression. Once the statistical evidence for association of genetic markers to a disease trait has been established, genome-wide eQTL mapping data can be examined to see if the same genetic markers are also associated with the quantitative transcript levels of one or more genes. Such markers are known as ESNPs. The availability of systematically generated eQTL information provides immediate insight into a probable biological base for the disease associations and can help to identify networks of genes involved in disease pathogenesis. The potential of genome-wide eQTL identification was shown originally in yeast and then in humans, animals and plants. eQTL mapping. In practical terms, the eQTL mapping is the measurement of gene expression in a target cell or tissue from multiple individuals. This information is the substrate for investigating the effects of DNA polymorphism on the expression of individual genes. The use of microarray technology to measure gene expression from many thousands of genes simultaneously has been a principal driving force for systematic mapping of eQTL. eQTLs are influenced not only by genetic polymorphisms, but also a range of other biological factors. These may be dissected systematically, starting with the measurement of heritability. Heritability. Family studies have demonstrated that many human eQTLs are highly heritable. GWA mapping of common genetic variants that underlie eQTL has recently become possible due to the wide availability of high throughput and low cost SNP genotyping. These information are particularly relevant to disease mapping that is also focused on common SNPs characterized with similar SNP arrays. Moreover, the interpretation of these eQTL data relies strongly on methodologies that have been developed for disease GWA. For example, a family study of lymphoblastoid cell lines identified nearly 15,000 traits with an estimated H-square of greater than 0.3, indicating that the genetic influences on gene expression appear to be widespread. Genetic factors with both cis and trans acting effects are often identified for eQTL that have high heritability. For example, in the LCL study mentioned above, eQTL for 81% of traits with H2 greater than 0.8 could be mapped to one or more SNPs at genome-wide significance. However, the SNP map on average account for less than 20% of the estimated trait H-square. This demonstrates the presence of genetic or other effects affecting familial clustering on transcription that are not detectable in these genetic associations. Cis and trans effects. Statistical analysis of eQTL need to take into account that the loci identified can influence gene expression either in cis or in trans. The definition of a cis effect is somewhat arbitrary, but cis acting eQTLs are typically considered to include SNPs within 100 kb upstream and downstream of the gene whose expression is affected by that eQTL. Detailed analysis of the position of mapped cis acting eQTL effects have shown that these are enriched around transcription start sites and within 250 base pair upstream of transcription end sites and rarely reside more than 20 kb away from the gene. Cis acting variants also appear to occur more often in exonic SNPs. In general, Trans effects are weaker than those in cis in humans and in rats, but are very numerous. It is not known if trans effects are generally mediated through transcription factor variants or other mechanisms. Master regulators are transacting factors with multiple effects of gene expression that have been identified in Saccharomyces cerevisiae, rat tissues and in the human genome. It is of interest that in yeast, master regulators are not enriched for transcription factors and trans-regulatory variation seems to be broadly dispersed across classes of genes with different molecular functions. eQTL and disease gene mapping, combining eQTL and genome-wide association studies. One of the most important consequences of eQTL mapping is the link that it provides between genetic markers of disease identified in GWA studies and the expression of a specific gene or genes. For example, a recent study generated genome-wide transcriptional profiles of lymphocyte samples from participants in the San Antonio family heart study and showed that high density lipoprotein cholesterol concentration was influenced by the cis-regulated VNN1, that is VNN1 gene. Similarly, a study of post-mortem brain tissue identified eQTL affecting the MAPT and APOE genes, which play an important role in Alzheimer's disease. At the same time as the San Antonio study, the results of a GWA study of asthma identified a series of SNPs in strong linkage disequilibrium and spanning more than 200 kb of chromosome 17q23 and strongly associated with the risk of asthma. The region of association containing 19 genes, none of which are obvious candidates to be implicated in disease. Examination of eQTL data derived from Affymetrix HU133A arrays on the same families showed that the disease associated SNPs had highly significant, that is p less than 10 inverse 22 effects in cis on the expression of one gene called ORMDL3. In these data, borderline significant effects were also seen in expression of the gene neighboring ORMDL3 GSD-ML. Subsequent eQTL studies with the Illumina platform and RT-PCR experiment confirmed that the same SNPs determined eQTL with both genes. These results focus attention on one or both of these genes as likely candidates for a role in disease pathology. Many additional studies are now underway to investigate the biological functions of these two genes and their relationships to asthma. Using eQTL to interpret GWA studies. Such findings have motivated the use of these eQTL data as a general tool for interpreting results from GWA studies. Recent analysis of Crohn's disease illustrate this approach. Initially, markers on chromosome 5 were shown to be strongly associated with the CD in one GWA scan, but their biological effects could not be readily deduced as they reside in a 1.25 MB gene desert. Examination of the LCL eQTL database showed that one or more of these polymorphisms act as a long-range cis-acting factor influencing expression of PTGER4, a gene that resides approximately 270 kb proximal to the association region. The homologue of this gene has been implicated in phenotypes similar to CD in mouse. Thus, research is now focused on PTGER4 as a primary candidate gene for this disease susceptibility locus. Subsequently, the eQTL approach has been applied systematically in a meta-analysis of GWA studies of CD, and several other interesting results have been obtained. For example, eQTL were used to address an outstanding question in CD genetics related to the identification of the CD susceptibility gene or genes in the cytokine cluster at 5q31, where SNPs have an established association with disease. The disease associated SNPs in the meta-analysis of this region were all shown to be correlated with decreased SLC22A5 mRNA expression levels. Another CD locus identified in the meta-analysis coincided with the asthma risk locus on chromosome 17, in which the disease markers are also correlation with the expression of ORMDL3 and GSD-ML. Thus, the same genetic variants contribute to susceptibility to both CD and asthma, possibly by perturbing expression of one or both of these genes. Examination of public's GWAS results identifies many other disease associations where eQTL data provides similar insights. For example, a recent large study of polygenic dyslipidemia identified 30 loci with highly significant effects on blood lipid measurements. Examination of gene expression in samples of liver from 957 subjects allowed highly significant eQTLs to be identified for 7 of the 30 loci. In some cases, the eQTL data gives genetic evidence to support a candidate gene for which a role was previously hypothesized from location and biological hypothesis, such as GNA12 for height on chromosome 7p22 and BLK and C8ORF13 for autoimmune systemic lupus erythematosus on 8p23.1. More often, the gene expression data identifies different genes or suggests a particular gene from a number of candidates. Examples of this are the cluster of trans-acting genes from the height locus on 7q21.3, the RPS26 gene from the type 1 diabetes locus on 12q13.2, and the DCTN5 gene from the bipolar disorder locus on chromosome 16p12.1. eQTL databases. A database of eQTLs from the asthma studies that allows searches by genes, chromosomal regions and SNPs illustrates how data from this kind of research can be examined. VerisysDB is another public database based on 1,90,000 extensively annotated mRNA transcripts from 36,000 loci. VerisysDB offers information encompassing published human genetic polymorphisms for each of these transcripts separately. In addition to SNP effects on transcription, this database includes deletion insertion polymorphisms from DB-SNP, copy number variations from database of genomic variants, short tandem repeats and single amino acid repeats from HLNV DB and linkage disequilibrium regions from DHaploDB. Gene mapping by human pedigree analysis. It is impossible to pre-select the genotypes of parent and set up crosses in humans specifically for mapping purposes. By examining the genotypes of the members of successive generations of existing families, the limited data for the calculation of recombination frequencies have to be obtained. Their interpretation is often difficult because a human marriage rarely results in a convenient test cross, and often the genotypes of one or more family members are unobtainable because those individuals are dead or unwilling to cooperate. For example, we are studying a genetic disease present in a family of 2 parents and 6 children. Genetic diseases are frequently used as gene markers in humans, the disease state being one allele and the healthy state being a second allele. The pedigree shows that the mother is affected by the disease, as are 4 of her children. The maternal grandmother also suffered from this disease, but both she and her husband, the maternal grandfather, are now dead. We cannot obtain any further information on their genotypes, but we can include them in the pedigree with slashes indicating that they are dead. For mapping the position of the gene for the genetic disease, we have to study its linkage to a microsatellite marker M. 4 alleles M1, M2, M3 and M4 which are present in the living family members. The pedigree shows inheritance of a genetic disease in a family of 2 living parents and 6 children, with information about the maternal grandparents available from family records. The disease allele closed symbols is dominant over the healthy allele, that is open symbols. The objective is to determine the degree of linkage between the disease gene and the microsatellite M by typing the alleles for this microsatellite M1, M2, etc. in living members of the family. The pedigree can be interpreted in 2 different ways. Hypothesis 1 gives a low recombination frequency and indicating that the disease gene is tightly linked to microsatellite M. Hypothesis 2 suggests that the gene and microsatellite are much less closely linked. The issue is resolved by the reappearance of the maternal grandmother whose microsatellite genotype is consistent only with the hypothesis. Let us summarize the following discussion on family studies and complex traits genetic maps. Gene mapping is the process of finding the location of genes on each chromosome. It is an important step in identifying the genes involved in a genetic disease. Once a disease gene is accurately located, we can determine its DNA sequence and its protein product. Relative to linkage analysis and sequencing, genome-wide association studies or GWAS have less power in cases where different rare mutations act in different families or individuals at the same locus. GWAS have been a resounding success, identifying thousands of disease-associated loci and revealing previously unknown mechanisms for conditions. Family studies have demonstrated that many human eQTLs are highly heritable. GWM mapping of common genetic variants that underlie eQTL has recently become possible due to the wide availability of high throughput and low cost SNP genotyping. Family studies have been used to identify monogenetic diseases, highly penetrant traits of biomedical importance, including a variety of inborn errors of metabolism and other genetic diseases. Before we attend the next session, please try to answer the following questions. Write an overview on gene mapping. What defines a family study? Discuss the importance of family study. Discuss eQTL mapping. Write a note on importance of SNPs. Here are some references for you. Introduction to Bioinformatics, Atwood and Parry Smith, Pearson Education. Bioinformatics, a beginner's guide by Jean Michael Claverie, John Willey and Sons. Structural Bioinformatics by Philip E. Bourne and Helge Willey. Bioinformatics, Methods and Applications, Rastogi SC, Mentorita N and Rastogi P, Prentice Hall of India Private Limited, New Delhi. I hope the session was enriching. Thank you for watching. See you next time. Till then, bye.